Epigenetic choreography. Unraveling the connection between DNA methylation and epigenetics. Introduction. In the symphony of genetic regulation, epigenetics takes center stage, orchestrating intricate changes that influence gene expression without altering the underlying DNA sequence. This video delves into the fascinating connection between DNA methylation and epigenetics, unraveling the molecular dance that shapes cellular destiny. 1. Defining epigenetics, beyond the genetic code. Epigenetics encompasses heritable changes in gene function that occur without alterations to the DNA sequence. It influences how genes are expressed and plays a pivotal role in cellular differentiation, development, and response to environmental cues. 2. The epigenome, a molecular tapestry. At the heart of epigenetics lies the epigenome, a dynamic collection of chemical modifications to DNA and histone proteins. DNA methylation, a prominent epigenetic mark, stands out as a key contributor to the orchestration of gene activity. 3. DNA methylation, marking the genome. DNA methylation involves the addition of methyl groups to cytosine bases within DNA molecules, often occurring at specific sequences called CpG islands. This modification acts as a molecular tag, influencing gene expression and contributing to cellular identity. 4. Enzymes and DNA methyltransferases, architects of methylation. DNA methylation is orchestrated by enzymes known as DNA methyltransferases. These enzymes transfer methyl groups to cytosines, modulating the epigenetic landscape and regulating the accessibility of genes for transcriptional machinery. 5. Role in gene regulation, silencing and activation. DNA methylation plays a dual role in gene regulation. Hypermethylation, the addition of methyl groups, often results in gene silencing by impeding the binding of transcription factors. Conversely, hypomethylation, or reduced methylation, can activate gene expression. 6. Epigenetic inheritance, passing on molecular memories. One of the intriguing aspects of DNA methylation is its potential for epigenetic inheritance. During cell division, the epigenetic marks, including methyl groups, are faithfully replicated, passing on molecular memories to daughter cells and influencing their developmental trajectory. 7. Dynamic nature of DNA methylation, responding to the environment. The epigenome, including DNA methylation patterns, is dynamic and responsive to environmental cues. External factors such as diet, stress, and exposure to toxins can influence DNA methylation, contributing to phenotypic diversity and adaptability. 8. Epigenetics in development and disease. DNA methylation plays a crucial role in developmental processes, guiding cell fate decisions and tissue specialization. Aberrations in DNA methylation are associated with various diseases, including cancer and neurological disorders, highlighting its significance in maintaining cellular health. 9. Demethylation and reprogramming, resetting the epigenetic landscape. Certain cellular events, such as cellular reprogramming and early embryonic development, involve active demethylation, the removal of methyl groups. This process allows for the resetting of the epigenetic landscape and the generation of pluripotent cells with the potential to differentiate into various cell types. 10. Therapeutic implications, targeting epigenetic marks. Understanding the connection between DNA methylation and epigenetics has paved the way for targeted therapeutic interventions. Epigenetic drugs, including DNA demethylating agents, are being explored for their potential in treating diseases characterized by aberrant epigenetic regulation. Conclusion. The intricate connection between DNA methylation and epigenetics unveils a sophisticated layer of gene regulation that extends beyond the linear sequence of the genetic code. From orchestrating cellular differentiation to responding to environmental cues, DNA methylation plays a pivotal role in sculpting the epigenetic landscape. Unraveling the nuances of this molecular dance provides insights not only into fundamental biological processes but also holds promise for therapeutic interventions that harness the dynamic interplay between genetics and epigenetics.